You cut it into multiple subsamples, and then you're going to run a regression on each of these subsamples using that lambda parameter we found before. And this will help to account for uncertainty in the data and also will prevent over-relying on a subset of the data for developing the final model. And then at the end, you're taking all of these different bootstrap models and you aggregate them together into a single model. So in these papers, and actually all papers did exactly the same, they made a conventional aging clock, which means you take all the proteins that you measure and you make a clock out of those. And then they made also 11 single organ clocks. So adipose, artery, brain, heart, immune system, intestine, kidney, liver, lung, muscle, and pancreas. And what they then did is they took all these proteins minus all the proteins they used to make the single organ clocks, and what was left over was called the organismal clock. So this is basically all the proteins that probably are important for other organs in the body, which were not these 11 organs that they tested. The study data here summarized. So you have 3 papers we are going to discuss. The first paper from 2023 used different cohorts of people that were studied. And here you can see the number of participants. In total, it was 5,666 people. These 2 studies used the UK Biobank, which has about 8 times more participants in total than these 5,000. So they are much bigger. The number of proteins here was more, almost 5,000 proteins. Here it's just under 3,000 proteins that were studied. Here this should be the same, but it's actually 7 proteins less because they excluded values for which they didn't have data in over 10% of cases, while they just used all the proteins. And here they use a different assay to measure protein concentration. We went over that earlier. So we come to the first study, the original paper in Nature from 2023. We pretty much went over all of that. So here you have samples, you have the different proteins, you have the age of the participants, and that was used to make that lambda parameter, which they here call alpha parameter, but it's the lambda, it's just another name. Then they used that in the bootstraps, as we discussed before, and then they aggregated the data together, and they came up with the predicted model. Here, predicted age, you can see chronological age, and you can see the correlation between the 2. And they did that for these 11 different organs, as well as for the conventional and the organismal, clock. One interesting thing is that they found that actually you only need a very small number of proteins to build such a model. So here you can see the number of proteins included in the model, and here you can see the predicted versus chronological age correlation. And you can see, like here, you're already getting at relatively very good correlation with small number of proteins. Another interesting thing that they found is that the proteins do not change linearly with age. So you can see there is a little curve in these curves. And that may remind you of a previous paper, this paper, where you remember that 3 different ages, you see a peak where lots of proteins suddenly change in expression. So this paper found exactly the same, that proteins do not change linearly with age, but there is sometimes a curve in it. So I found that quite nice that they found that. So what they also found is that 23% of the studied population are extreme ages, and don't mistake yourself, this can also mean that they are younger. So not just older, but extreme in the sense of being deviating from normal. Here you see the 5 different cohorts that they studied, and you can see most of the cohorts all show just over 20% of extreme ages, except this one cohort, which was a cohort of normal cognition. I think this was a small cohort that only showed 10% extreme ages. This cohort, the longevity, is from Nirbacilli, and that is on people who are very exceptionally long-lived. And their offspring, interestingly, they also show about the same amount of exceptional or extreme ages as the other populations. But what they didn't show is the percentage of here who are accelerated aging versus decelerated aging. So we don't know that. But when you look at this graph, you can actually see that there are very few blue lines. The blue lines is people who have slower aging, and the red is faster aging. So in almost all the cases, people are aging faster for a very specific organ than on average. And what you can also see that is interesting is, so these are all individuals. Here is individuals, and here are the different organs that are aging. And for example, here you see a whole group of people who have accelerated heart aging, but they don't show any accelerated aging in any of the other organs. And the same you can see here, for example, lung, not in any of the other organs, kidney, not in any of the other organs. So in fact, if you look at it, there are actually only a very slim number of people who show aging in more than one organ, accelerated compared to the average. And yeah, extreme ages were 2 standard deviation below or above the mean. Is what was here defined. Later we will see where they switch to 1.5 standard deviations in another paper. Individuals with the same conventional age gap can still show an accelerated aging in a specific organ. So here you have someone who has kidney aging accelerated, but is completely normal for all the other organs. And here you see someone who is accelerated in many organs. And so here you see people who have the same conventional age. This person has accelerated aging in a lot of the organs, while this person has actually decelerated aging in many of the organs, despite them having the same conventional age. So these single organ age scores do measure something that conventional age is not able to capture. It's also interesting that there is little correlation between different organ ages. So that means you can have very aged lungs, but that doesn't mean you have aged arteries or aged intestines and so on. There is little correlation between that. And so here, a few people, again, this is a little bit of repetition, of multi-organ ages. In fact, you can see there is only like 1.71% of people, so less than 2% of multi-organ ages. So you can see the red dots, that's all the organs in which they were aged. Here you have someone who is a kidney ager. There is 2% of the population there, and you see they are not aged in any other organ. Here, heart ages, not in any other organ, also about 2%. So in fact, for most of these organ systems, we see like about 2% of people are aged in a particular organ. And this extreme aging in an organ is correlated with major age-related diseases. So here you see the clocks, and here you see the diseases like diabetes, for example, and you see people with older kidneys, they have a much higher risk of developing diabetes. They also have a much higher risk of obesity, hypercholesteremia, and hypertension, for example. People with older hearts have higher risk of heart attack and arterial fibrillation. Arterial fibrillation is also a heart disease. It's an abnormal rhythm of the heart. Here, one thing that absolutely will not surprise anyone is people who have old muscles will also have gait impairments, because your muscle is, of course, important for your gait. So you can see clearly that the organ clocks are predicting specific diseases. And so here you see, for example, this is kidney aging, these are heart aging, and people with older kidneys, they have also, so if you have 1 year older kidneys, people who have hypertension, I should say it like that, have 1 year older kidneys. People who have diabetes have 1.3 year older kidneys. People who have arterial fibrillation have 2.0 years of accelerated heart aging, and people who had a heart attack have 2.6 years of accelerated heart aging. So again, diseases correlate with older ages of the organ.
Sven, could you go back to the previous slide for a second?
Yeah.
Okay, so let's look at diabetes versus lung, and we see that it's nicely blue. What's that really tell us?
Yeah, people who have younger lungs seem to have a higher risk of diabetes. There are not that many blue things, as you can see. So there are not that many cases where an organ is younger in a particular disease. It's mainly the opposite. There is no star there, so it probably means that this didn't reach significance. Right, so none of the blue ones, as you can see, have a star in them, so none of them reached statistical significance. So this might just be something like, pure random luck. Additionally, I don't think that diabetes will make your lungs get younger, right? But there might be some other factor that causes your lungs to be young, but that also happens to give you diabetes. Like maybe a lifestyle choice that is maybe good for the lungs, but bad for diabetes risk. By the way, any other questions at the moment? Or can I continue? Oh, I see messages, anything?
David made a comment about the use of the term extreme when it's 2 standard deviations. That's actually, that's like what, 5%?
Yeah.
Is outside of...
I took over the terminology that the authors used, obviously. So if they call it extreme, then I call it extreme because I'm just presenting what they do. But yeah, you can fall over that word choice, extreme or not. I see, very interestingly, Albert, who points out that low carbon monoxide is good for the lungs, but it actually increases diabetes risk. So maybe this is already one explanation here. Yeah, so here in this slide, you can see the organ age gaps, and here the risk for heart failure, and very obviously heart failure with heart aging, but also some other organs when they show that if you have older age in that organ, you have a higher risk of heart failure. And for heart aging, this is actually 23% increase for every year of heart aging. And so here we are looking at all-cause mortality, and you can see a lot of the single organ clocks, or either conventional and the organismal clock, all are showing a positive association with all-cause mortality. And in fact, what they find here is that one standard deviation increase, which is about 4 years in organ age, is associated with a 15 to 50% increase in all-cause mortality.
If I may ask a question, how did the scientists account for extreme interventions, say a kidney transplant or you name it, or that they were excluded?
I don't know exactly how that worked. You really have to look then at the inclusion and exclusion criteria. I know there was a paper where I read something, but I don't think it's opposite in this one. For every single cohort, I think it was this paper where they have multiple cohorts. For each cohort, they list, the inclusion and the exclusion criteria, I think. Yeah, they had exclusion criteria as well. For example, if you had uncontrolled hypertension, if you reported yourself that you had cancer at the time, except for squamous cell or basal cell carcinoma of the skin. If you were pregnant, you were excluded here. If you had a chronic infection at the time, you were excluded, and so on and so forth. So yeah, you will have to look at the criteria. I don't know them on top of.
Notice that there's a couple questions in the chat window as well.
Drivers of self-destruction or the responses to damage, something else. Well, for the first, I think they probably are both. I think that gene expression changes with age can be both drivers of aging, but they can also be a response to aging processes where the body is trying to maintain homeostasis. So if you have an increase in oxidative stress, you for sure will have an increase in the expression of superoxide dismutase catalase and so on. But those are not driving aging. They are trying to counteract it. So your body is responding to the damage. So I think you will both.
Can I follow up, Sven?
Yes, sure.
If there is this distinction, yes, I've been writing lately about this distinction is very important. these gene expression changes that are driving aging, we want to minimize them, but the gene expression changes that are responses to aging, we want to increase them. If I score older on these responses to aging, it means my body is trying to rescue itself better. And distinguishing between these two kinds of changes seems to me essentially important. And constructing a single age measure that combines the two seems to me self-defeating. Some of these gene expression changes are good for you. Some of them are bad for you. And to combine them in a single clock seems to me to be missing the main point of what we want to be doing with these clocks.
I think that's a good point, but I think that distinguishing between the two is pretty much impossible. We don't
It's not
know
impossible,
exactly.
it's just hard. I've written about ways that it can be done, but I think it's absolutely essential. Combining the good changes with the bad changes into one clock doesn't tell us anything.
Possibly, yes. That's a good point, yes. That's indeed why we have to definitely take these clocks with care. I see David has a question too.
Just a response. I think Josh makes a good point that there are two different types of gene expression. But from the point of view of these articles, the point is to provide a fairly low-cost method of indicating that organs are aging. And both types of gene expression will indicate that organs are aging, and therefore people may want to change their lifestyle or have some treatment in order to avoid having more changes in these organs. So philosophically, and for other reasons, it's important to make that distinction. If all we're trying to do is get a cheap and effective way of alerting people that there are issues in their organs without having to drill into each organ separately, these findings are important in both cases.
So David is indeed correct here in the sense that if you see a response of an organ against aging, like the thing I already mentioned, the SOD increase, for example, or the catalase increase, that means that the organ is under stress. So it is also, even though it's contracting aging, it's also measuring the fact that the aging is under, the organ is under stress. So it is capturing, in that sense, the aging clock. Your clock may then be very good at looking at people and saying, oh, your heart is aged or your liver is aged, but the clock may then fail when you start to do an intervention that improves the aging of the organ to that then capture that correctly, because you're also looking at intervention in things that are protective. And so then if you increase those protective responses, the clock will say your organ is aging faster, while actually your intervention is protecting against the aging of the organ. So
Yes,
yeah,
thank you.
sense,
That's exactly what I wanted to say. Thank you for saying it so clearly.
Right. So if I have COVID, I might expect to see my numbers for my lung go way up.
that is possible, yes.
Because my body's responding to the infection in my lungs.
Yes, that is indeed possible. And then, yeah, the clock may not capture any type of intervention like rapamycin or whatever that will be beneficial in preventing aging of an organ.
Did you see Rick's question? What phenotypic biometric correlations were used to confirm protein markers to validate?
I think we will come to the fact that the clocks will predict, for example, your risk of Alzheimer's disease and things like that. They didn't look at individual proteins so much, so they didn't in the paper.
Muscle test.
No, they didn't do any muscle testing or anything.
Let me interject. Yeah, so as a engineer physician, at the end of the day, working in a translational clinic, we can look at these proteins and genetics and we can postulate what's important. But my engineering perspective is like, how does that improve the function of the human? And not concerned about pathology, concerned about age resiliency and function. So when I do a protocol, I want to see reaction time. I want to see ejection fraction improve. I want to see vascular integrity improve. You know, we don't necessarily have to look at everything, but it would seem to me that we could pick some real key functional markers that we could say, we do an intervention, we see these protein markers change, and then we can validate it with a functional change in that organ system. And then we can have a correlation of something is improving, something is getting younger, and not getting into the weeds of what is our percent risk of pathology reduction 20 years down the line. You know, I want to know how and what I'm doing is affecting someone next year, 6 months from now, 2 years from now, because I'm not concerned about 20 years from now, because if I'm doing the right thing for someone, they're never going to get to that pathology. It's that vision, like even I'll stop there, but that would be really important to pick a few systems, because then we could not only track people at home, we could track people more regularly, and then we could validate with the protein markers, because blood age markers, as we're discussing, are confusing, they're expensive, they're slow. Like, do you do, forgetting epigenetics, true diagnostic, glycan age, right? We can telomics, you know, there's 17 of them you could do. And most people do them once, they do them randomly, and they just don't know what to do with them. So what excites me is having a validated marker that we continue to track to verify and validate the things that I can see on a regular basis. And that way we can triangulate and really make big changes, especially when we start getting data and analyzing them with AI. If we could do that, that would be awesome.
Okay, they didn't really look at any type of phenotypic measurements. What they did do, I don't know. And now I'm going to have to scroll a little bit through the presentation. Probably it's in a later paper. Where is it? Yeah, here. So here actually they look at all-cause mortality risk and they show that the aging, organ-specific aging clocks adding towards pheno age as a predictor. So they can improve upon pheno age by itself. So there is, these clocks may capture some functional work as well. They haven't really looked at any functional outcomes yet.
That'd be great to do. Okay. Yeah, I'd love to, you know, even pick a few markers and see if we can validate those. I will reach out to you.
Here they used a special algorithm, FIBA, where they looked at the brain specifically and they tried to make a model that could be even better than the previous one at predicting brain aging. And they found one. And so here what you see is this is the global clinical dementia rating. So it's very well predicting if someone has dementia, yes or no. So that's, cognitive tests, right? And then here you use their new brain aging clock, and their brain aging clock is capturing very well if someone has dementia or not, much better than, for example, the plasma tau levels, age, or any other things. And when you look here, this clock, and here is time over years, and you have people who have low brain aging with the risk of them developing dementia versus people who have high brain aging, and people who have high brain aging have a much higher risk of developing dementia compared to people who have low brain aging. And you can see it stratified for the different, every step here up is a higher, people with higher level of brain aging. So the 0 will be the people who have the normal ages, and the minus, the blue lines, are the slower aging ages, and the red is the faster ages. So then we come to the second paper. So this is the neuroscience paper. So that's from a different group. The third paper is again from the same group as the first paper, but I put it in chronological order of which individual papers basically were published. Here they looked at the UK Biobank, and the third one will also look at the UK Biobank. And remember, here we have about 8 times more people than in the previous paper, so we have much more robust data. So here they find quite a lot of proteins go up with age, much less go down with age, as you can see. And then they looked at mortality here, and the brain aging is the best predictor of cause mortality, and followed by whole blood, pancreas, and so on. And then here you can see no organ aging, the survival probability 15 years of following up, and you can basically see that's here. But if you have 3 or 5 organs aging, that's the survival, 6 to 8 organs, and here over 8 organs. So the more organs that are aged, the worse your survival probability becomes. I don't think anyone is surprised by that. But the brain, yeah, as I said, that's the one that is the best predictor. Very interesting is the heart is actually not such a good predictor, and it's actually just as good as your skin in predicting mortality, which is very weird given how many people die of heart disease. Here, they looked at some factors like chronological age, sex, smoking, education, BMI, and physical activity. And you can see in green is 3 to 5 organs aging, blue is 6 to 8 organs aging, and red is over 8 organs aging. And when you get older, when you look at older people, the number of people who have 3 or more, I should say, sorry, organs aged increases. When you look at male versus female, you see that males have more organs aged than females. When you look at smoking behavior, people who smoke have a lot more organs aged than people who are not smoking. This education is, by the way, educational deprivation. So these people are better educated than these people. It's a little bit the opposite of the other measurements here, because it's deprivation, meaning that if you have higher deprivation, you indeed show a higher proportion of the people have organs that are aged. BMI, again, you see this, except for the people who are underweight, but of course here you are probably capturing people who are chronically ill already. That's why you probably see this. But with obesity, you have an increase in the organ age. And physical activity. So these are the people who are least physical active, yes. So they have a higher proportion of organs that are aged compared to the other ones. But what is interesting is that between these groups, there is no difference. According to this, you only need a very small amount of physical activity. But that is not what we see when we look at mortality. For example, we see that mortality keeps decreasing with higher physical activity. So here the clocks do not match the data that you get from epidemiological studies.
Can I insert a comment here on
Sure.
this page? You said earlier that it's impossible to tell which are the beneficial changes and which are the detrimental changes in gene expression that change with age. And I would suggest that we have right here good examples of one way we could tell. We suspect that smoking ages you not by changing your gene expression, but by damaging your lungs. So if there's gene expression in response to smoking, it's probably good. It's probably the protective gene expression. And those should be counted as what I call type 2 changes in age that are good for you. On the other hand, if things change with physical activity, that's likely to be type 1. Physical activity changes your gene expression in ways that make you younger. So looking at these two different profiles, we can start to develop hypotheses about separating the beneficial changes with age from the detrimental changes with age. For example, the changes that occur with physical activity are presumed to be type 1. They're detrimental changes. If you're older, according to the physical activity clock, then that means that that's reliably telling you, yes, you're really older. And if you're older according to the smoking behavior changes, then that's likely to say, count that as younger because you have more of these rescuing phenotypes that are rescuing genes that are being turned on.
So you say that smoking will not change gene expression. I disagree with that.
I'm saying smoking changes gene expression indirectly. It damages the lungs, and in response to that damage, there's gene expression change. And so that gene expression change is likely to be rescuing. Is probably the kind of gene expression change that we want.
Some of it, but I don't think everything. Like if you smoke, you have a high increase in inflammation. And I think the inflammation here is not going to be beneficial. It's a response to damage. You know, not all responses to damage that our body do are beneficial. Evolutionarily, we have never learned to deal with smoking, right? So our body perceives damage and it probably responds to that much more like it was an infectious disease, so it boosts inflammation. But of course, you keep smoking for your whole life, so the inflammation doesn't clear, and inflammation is trying to deal with infection, but there is no infection, so this is not beneficial. I don't think that you could say that smoking is beneficial gene expression changes you are seeing.
Can I come in on the question of smoking? Although I'm not a fan of smoking, I've never smoked myself. It's known to be protective of Parkinson's disease, and obviously it creates mild hypoxia. And creating mild hypoxia does change gene expression. So it's a complicated one. There are positives and negatives. I think the negatives outweigh the positives. But I don't think you can say it doesn't change gene expression because it does.
Thanks to both of you. Those are very clarifying for me and helpful.
I believe people who smoke are less likely to die from COVID, if I'm not mistaken.
The other thing about smoking is that it has nicotine in it, and it triggers the A7 subunit of the nicotinic acetylcholine receptor, which is our key endogenous evolutionarily conserved receptor for inhibiting inflammation. I think it's the nicotine because, again, nicotine pings the A7 subunit of the nicotinic acetylcholine receptor.
Here we look at the different age organ gaps versus different diseases. People who have older lungs have a higher risk of, for example, respiratory system diseases. Yeah, that wasn't surprising. They have also a higher risk of musculoskeletal system diseases. That's maybe a little bit less suspected. So here, the brain, for example, the brain is only associated with mental and behavioral disorders and nervous system disorders. So that's a bit surprising. The brain is not correlated to any of the other. Sorry, I should say these diseases are not correlated to any of the other clocks. I should say it like that. And here, that's the same, but it's for the different types of cancer versus the aging clocks here. And then we come to the third study, and the third study is again using the UK Biobank by the same authors as the first study. So they expanded on their first work. What they find is that the aging organ gaps are sex dependent. So kidney, immune, and intestines tends to be older in males, while heart, artery, and adipose tends to be older in females. What they also find is that the conventional age clock, the one that uses all the proteins, misses most of the organ-specific signatures in the sense that if you look here, organismal, there's a clock based on all the proteins minus the organ-specific ones, accounts for 74% of the conventional aging model, which is based on all the proteins. So it means that the individual organs themselves are only a slight fraction of that, and most of that is the brain aging and the artery aging. So, for example, heart aging, pancreas aging, lung aging clocks, they are not really in here at all. The aging of these organs are missed by the conventional aging clock, basically. So here it's something a little bit similar to what we have seen earlier, but there was more data where we saw that most of the people who had extreme aging were less than 2% in a single organ. But here the age gap is only 1.5 standard deviations, or even less extreme than in the previous one. And I already got called out for calling it extreme there. So these are mildly increased, let's call it like that, mildly increased. So 27% of the population were normal agers, 14% of the population had both increases in aging in a specific organ and decreases in other organs. Here you can see this is people who have multi-organ aging of 2 to 4 organs. And these are people who have negative, so these have where they are in 2 to 4 organs less aged than average. And you can see that's over 10% of the population, and then all the rest is below 2%. Interestingly, people who are above normal in a single organ, they tends to be older, and that's true for people who are positive or negative ages of that organ. So here you have normal, and here you have people who are less aged in one organ, less aged in 2 or 4 organs, less aged in 5 to 7 organs, and here more aged. And you can see that in both cases, the people are chronologically older. Organ age gaps remain stable over a 1 to 15-year follow-up of the baseline. So here you see when people's organ age was measured at the first visit versus the second visit, and it correlates for all these different organ systems. If you are old in an organ at the first measurement, you are still aged a few years later in that same organ. You don't just rejuvenate spontaneously. And that you see here as well for the people who are more old and the people who are younger. You see if you are very old in the arteries at the first visit, you are still very likely to be very old in your arteries at the second visit. You don't show much rejuvenation. So here we look at future disease gaps with the organ aging. So here you have the different organ age. Here you have the different diseases and the correlation between them, which is very similar to what we discussed before. They categorize the illnesses slightly different. So I can't really see if these correlate very well because it's a different classification of illnesses. Here they put them in categories like cancers, infectious disease, and so on, while here they go to very specific diseases like heart failure rather than calling it heart system or cardiovascular system.
Sven?
Yes.
What about something like RDW where there's a spread well associated with bad outcomes? If you have a high red blood cell distribution, it doesn't seem to bring blood into the picture at all.
No, they haven't looked at any of these typical measurements or
Okay.
clinical measurements. They haven't done that. So there is no data on that in the papers.
Okay.
This is again capturing something I have told before, is you have certain people who have systemic aging, so many organs are old. And that's true for people who suffer from chronic kidney disease, heart failure, or COPD. They have aging in many organs going on, while people who have, for example, Alzheimer's disease, they have very old brains, but not much aged in the other organs. Atrial fibrillation, a type of heart disease, they have old hearts, but not other organs aged. People who have type 2 diabetes are old in the immune system, in the kidneys, and in the intestine. And interestingly is that tissues like the liver, the pancreas, the muscle that are important in diabetes do not come out here. That was a bit surprising to me. Okay, oops. Here, the brain age gap predicts Alzheimer's disease risk. So here you see the different organ clocks for Alzheimer's disease and brain pops out. Conventional pops out, so all the proteins. Organismal is here, that's all the proteins except for the ones for the single organs. And then all the single organs. And so you can see it's mainly the brain age and the conventional ones that will predict your risk for Alzheimer's disease. And so what you see here is people who have a normal brain, people who have an aged brain, and people who have a youthful brain, and their risk to develop Alzheimer's disease over 15 years of following up. And you can see that people who have youthful brains almost never develop Alzheimer's disease versus people who have old brains who are at much higher risk.
Looking at lungs in the first graph, it almost seems to say that having a good set of lungs may serve to prevent Alzheimer's.
I don't know if that reaches statistical significance. I
Okay.
have no clue on top of that, of my head for that. But potentially, yes. It's very similar. What is here interesting is here, people who have 2 to 4 organs that are aged more than average have a higher risk for all these diseases. So that's kind of what you predict. If you have multi-organ aging, you really are bad off. Here you have people who are aged 5 to 7 organs. And again, you can see that they have a much higher risk for all these diseases, particularly here for chronic kidney disease. Also very much here for heart failure. One thing that is more surprising is here, people who are younger in 2 to 4 organs. Yes, you can see some blue, but not that much. So they are not that well protected against diseases. Here, if you look at people who have 5 to 7 younger organs, they are only protected against ischemic heart disease, but they actually have an increase in type 2 diabetes and in Parkinson's disease. And this is statistical significant. So people who have very young organs have higher risk of Parkinson's and type 2 diabetes. No idea why that is the case.
That could actually support Josh's contention a few moments ago that their responses are very effective.
Yes, you're looking here at protective. So here, this is progression over time versus the aging gaps. What it shows here is that, for example, for kidney disease, when you go after time, this time after diagnosis, so when time goes on, the pancreas, here you are looking at pancreas age gap keeps going up. So your pancreas gets older and older and older. In people who have this kidney disease, but not in healthy people. But if you look here at dementia, it stops doing that. What they are basically saying is that the brain age clock is predicting if someone is going to develop dementia. But once you have dementia, it doesn't predict how fast the dementia is going to go. But that makes it, of course, interesting if you can predict the dementia before it occurs. That's much more interesting than the progression rate of dementia, probably. So that's maybe a good thing. And what you see here is that chronic kidney disease is really linked to all of these aging clocks here very strongly. And for the rest, it's actually not so much correlations happening. Here we look at lifestyle, drugs, and supplements. It doesn't surprise you that smoking is really bad for you, and many of the organ clocks are getting accelerated in aging. Alcohol doesn't seem to be so good for you either, but things like fish oil, so oily fish, vigorous exercise, poultry, moderate exercise, some beef, pork, this probably is no longer statistical significant here. These are things that are good for you. And here, some other things that are bad for you, processed meat, bread, and so on. Here you see those were for drugs and supplements, Premarin, ibuprofen, glucosamine, got liver oil, multivitamins, and vitamin C. And you see they all decrease, for example, the kidney aging clock. Liver aging is significantly here by Premarin, reduced and so on. Women who have premature menopause have accelerated organ aging. Here you can see the aging in all of these different organ systems in women who have premature menopause. And when you treat women with estrogen, you see decreases in these different clocks here. And that you see here as well. If you give estrogen to people who have here normal early menopause or premature menopause, you always see that there is a decrease in the immune age clock. That's this one here, which is very strongly increased in people with early menopause and is strongly decreased with Premarin supplementation. So Premarin, a type of estrogen. This one we briefly touched upon earlier is showing that here you see the pheno-age line, and you can see that, for example, if you use the brain aging clock that I developed, you can get above pheno-age. These clocks give you information that you will get from pheno-age alone in terms of all-cause mortality. Here you see people who have extreme accelerated or youthful aging and the risk for mortality. So people who have accelerated brain aging, they show a higher all-cause mortality, lung aging, and so on. And here you see people who have lower aging, so lower artery aging, immune aging, brain aging, and so on. Here you see decreases in mortality risk, so they are better off. But for example, here you see that people who have young arteries are actually accelerated agers. And the authors mentioned that the reason for this may be that this specific protein that was very important for the artery clock had a big weight to it, is actually decreasing with age, but it's positively associated with mortality. So that comes a little bit to what Josh was talking about. If you look here at normal agers, or people who have many organs in which they are aging, or here people who are low agers on the brain and the immune system. And then you see if you are low ager on the brain and immune system, you have very low all-cause mortality. Here is a normal. And then when you go down to more and more mortality, it's with an increase in the number of organs in which you have aged. Here, what is interesting also is that people who have younger ages in multiple organs do not seem to have a decrease in all-cause mortality, which is surprising. So the papers also have some limitations. So far, not all of the organs have organ-specific clocks developed, and so we don't know of if we can make such clocks for all the organs. Might be that some organs just cannot be accurately predicted their aging in blood. The clocks were mainly trained on data from older people, and because proteins don't change linearly with age, it is possible that these clocks might not apply very well to young people, and that's why we should also be looking at non-linear models to make such clocks. And there is a lot of American and Caucasian skewing in these cohorts, so we need to look at other populations. I haven't actually read the paper yet because it just came up to me when I was doing some research for this journal club, is that there is a new paper in Cell Metabolism that is also further building upon this work from Vadim Vladishev. So you might want to read that, and that's the end of my presentation. So questions, remarks? I see that Albert has a question on green tea, black tea, or both. I don't know which that was. Maybe that's listed somewhere in the method section of the paper, but I don't recall if it's what type of tea they looked at.
These are mostly people in the UK, so I'm thinking it's mostly black tea. They couldn't possibly have a majority drinking green tea there. Well, maybe they could. I shouldn't say that.
That's a good suspicion, yes.
Another possibility is that people that are drinking tea are putting a lot of sugar in their bodies on an empty stomach or something like that. It might be a sugar or cream effect.
Yes, yeah, that was another thing that I also considered that might be possible.
Can you give your personal opinion since you took such a deep dive here? Now that you've come up for air, this was a fabulous review. Are you convinced that this is the way to go in the future, or do you still see a lot of problems and we shouldn't give up the current aging clocks that are based on whole animals?
I see that these clocks have potential, but they are very new, only like 2 years old or so. So I think there is a lot of research that needs to be done. And as was pointed out correctly in the journal club, we have not correlated these with, for example, phenotypic measurements. So it will be very interesting in future work to see if accelerated muscle aging, for example, correlates with grip strength and things like that nature. So a lot more work needs to be done also with, like Jos pointed out. Maybe more interestingly would actually be if we can use interventions that extend the lifespan or improve the function of an organ and then check what the aging clock does. So rather than trying to, in a sense, pull apart the proteins that you use to make the clock, let's start with the easier thing, and that's actually to just see if you can do an intervention, improve the health or the lifespan, and then see if the clock actually goes back or if the clock will accelerate. If the clock will accelerate, you will actually have proven just his point that you are actually measuring protective responses. So that will be an easy type of thing. And actually that reminds me on this, there was this methylation age paper out there not so long ago. And for example, it showed that rapamycin accelerated multiple methylation clock ages. And so this might be actually an example of that.
Do you think your paper supports the idea if you try to keep your brain young, it may actually keep the rest of your body young too?
It might be. Now, again, none of these papers were tested on interventions, right? So it may just be that if you have the lucky genetics, for example, that you have younger brain, then that results in also lower aging of the other organs. So you can't really exclude that possibility. But I would assume that there is a good likelihood, yes, that if you keep your brain healthy, that other organ systems are indeed also younger. And we do know that, for example, in mice, we do know that you can actually have an impact on whole body aging by influencing just particular sets of neurons in the brain. There were some papers on that a few years ago. If you had like a manipulation in NF-kappa B signaling in particular parts of the brain, you could actually control whole body aging.
If I may comment about Josh's point, Let's say you have, we'll just use COVID as an example, since that's the only thing people ever catch anymore anyway, right? So you would have a period where you'd see these proteins in your blood. So that would mean, your lungs are trying to protect themselves. And then you would expect that if the disease receded, the proteins would also recede. But if it goes too far, you just simply die. It's kind of a two-way street all the time, reflecting response, but it's also reflecting the level of damage, because presumably more damage would be more response.
Remember, Walt, there was only one pathway, the arterial measures, that were correlated with an increased risk when it looked like they were younger. And conversely, almost every organ that was showing the aging markers was also correlated with increased risk of death from a disease in that organ. So there could be some benefit, but these results overwhelmingly suggest that the body's response is insufficient, and the more it cranks up that looks like aging, the worse the outcome, not the better.
Good point.
Walter, you're muted.
I was congratulating Sven on a wonderful job. He's a lot of work put into this, I'm sure.
Thank you, Walter.
48 pages, that says a lot.
Yes, the people of you who have read the papers know that it's very dense, the papers. And then we had so many supplementary and additional extended data figures. So not only supplemental, but extended data figures. So quite a lot of material to go through.
I see that Steve has put something up.
I'll just briefly say what I think. There's that great James Burke documentary of decades ago now. It's called The Day the Universe Changed. And what it had to do with was some discovery that changed the way individuals and people thought about a problem. And I think the Day the Universe Changed study of 2024 was published on April 16th. I've posted a link in chat. And essentially, some folks in China did a masterful study related to young blood. And they discovered that extracellular vesicles in young blood had a few microRNAs that ended up triggering increased PGC1A, and then they oriented their experiment to determine whether increased PGC1A was essential for achievement of the Younging effect, and it was. In their experiment, they found that increased PGC1A expression was a necessary condition to achieve the Younging effect. And so when I saw that, I thought, well, if this is true, I'm not saying it's true, I think it's true. If it's true, this is a day the universe changed. I have spent the last 4 or 5 months exploring this issue. You'll remember that in 2022, August, the convoys discovered what the intracellular pathways were for aging plasma, that aging plasma was targeting. And now we have variables, features that are associated with young blood. I think this sets us up in a way we've never been set up before, where we're not just talking about at the intervention level, right? Young blood, aging blood. We're talking about the mechanism of action level. And I've posted a list of the interventions that trigger PGC1A. It's an astonishing list. I encourage you to look at it. There's a study called PGC1A is a master regulator of mitochondrial life cycle biogenesis. What that study does, if you take the whole breadth of these studies, what it's demonstrating is that young blood experiments have positive impacts because of impacts on the mitochondria. PGC1A increases ATP, it results in longer telomeres, reduced senescence, better gut health, and the longer telomere experiment, along with reduced senescence, that was demonstrated in a hyperbaric oxygen experiment, not just young blood. So I think PGC1A is the independent variable we ought to be focusing on, because if these folks in China have figured out something that is a day the universe changed, we can all benefit. Sorry for taking up so much time.
No. Could you remind us how PGC1 is increased? How do you do that?
I made a list in the chat. Young plasma increases it.
Oh,
Hyperbaric oxygen.
oh, I see it. Okay.
Cold plunge, PEMF. Have you folks seen that Halo device? The PEMF device called Halo? My wife and I just got one. She'd been diagnosed with collagenous microscopic colitis. Day one using Halo, symptoms gone. Use it every day. Colitis symptoms gone. PEMF triggers PGC1A, and PGC1A has been shown to specifically address colitis. It's astonishing. I'm excited about this, and we need you, Josh, to take a look. I think we've turned a corner here.
You say a few more words about Halo?
Yeah.
Okay.
I'm about to initiate a contest, and the contest is, I'm serious, I'm going to do this. This is the first time announcing it. Sonnet 3.5 has helped me to draft the contest rules, but essentially the rules go like this. Name a profoundly positive intervention that doesn't either increase PGC1A, in other words, increase the factor in young blood, or inhibit NFKB, in other words, inhibit the factors the convoys found were critical. Find a profoundly positive intervention that doesn't do one of those two things. If you can, and I'm not going to do this until I publish the contest rules, because there are some exceptions, I'll give you $50 of Bitcoin. What do you think, Walter?
I want to enter the contest.
Well, there we go. I want everyone to enter the contest too.
Can you clarify, are you suggesting that PGC1A is associated with longevity when increased or decreased?
PGC1A, except in cases of cancer, Cancers, there are paradoxical effects. Okay, so literally I've spent 3 or 4 hours working with Claude Sonnet on the rules for this contest. Cancers excluded, mechanical interventions like cataract surgery, arthritis surgery, that kind of thing. Those are excluded. But PGC1A is essential for mitochondrial biogenesis, and it does a lot of other positive things. And we know that NFKB inflammation is underlying a whole lot of these diseases. And get this, PGC1A and NFKB, I'm going to put this in slang. I'm certain I'm not going to express this in a completely technically correct way. But in the nucleus, NFKB and PGC1A struggle for control of the P65 telomerase subunit. Do a Google search, PGC1A, P65 telomerase, and NFKB, you'll see it. So either I'm way out to lunch or the universe has changed.
So here, this was one example of a paper where they found that specifically within the hypothalamus, systemic aging was being coordinated. Here it's in C. elegans, 2 neurons that mediate dietary restriction-induced longevity. Here is again in the hypothalamus, mTORC2 controls metabolic health and longevity. Hypothalamic menin regulates systemic aging and cognitive decline. So you see there are multiple papers showing that the brain is some type of central regulator of lifespan and aging. And that's exactly what we also see here, that brain aging specifically was the best predictor.
Yes. Okay. Well, that was a wonderful meeting. Is there anything more?
What's this Halo machine? I mean, is it worth buying? Like, should we just drop everything and go buy one?
$3,000.
Whoa!
Is it an electromagnetic field generating device?
Yeah, it's electromagnetic field. It's a big device. We have the device, the power unit, sitting. I'm going to get a picture of this soon. Sitting on top of the dog crate at the end of our bed, and we have the cord for the Halo device, which weighs 5 pounds and is like 8 inches across. And my wife and I are exchanging it throughout the night. She uses it morning and night for her gut. She's used it on sinuses. It took care of sinuses. I've used it on my skin. I can feel the increased collagen on my skin when I use it. I use it for inflammation, and it's quite an amazing device.
Have you done anything to independently measure the strength of the EMF meters, and if it has a scale of any kind, what's the range of power or intensity it suggests it's giving the user?
So I've spoke to Halo technical support about this issue multiple times, and they do not provide information about the intensity of the signal or what the frequencies of the signals are. But because what they're doing, they say, is they're putting together scripts of PEMF frequencies to address problems, and those scripts are proprietary. What I do know is I put this thing on my brain when I want to wake up, and it takes about 5 minutes and I'm awake. It's pretty great.
I urge somebody to rent a tri-field meter for $100 from a tool supply center and measure it. It
Right.
shouldn't take long. How many settings are there on the device? Is it like low, medium, and high, or like 15 different settings?
It's more like they have a menu of conditions that you may want to treat. But we just got this 2 months ago, and we realized that having this cord up the middle of the bed so we can exchange it through the night is going to be a lifetime habit now.
Yes. I guess one thing that is a little bit of a concern, how will I even know it really works if they can't even tell me what it is?
Because you'll rent a tri-field meter and measure it yourself.
Right. Well, you could actually just set up an antenna and a frequency analyzer and probably get majority of what you need.
I bugged them about this, and I'm going to bug them again, because another way that 2024 saw the universe changing, 2 different studies, one at MIT and one in China, demonstrating that 40 hertz triggers the glymphatic system to process brain waste. And 40 hertz has been shown to reverse cognitive decline.
Now, wait a minute. I thought I saw that 40 hertz is where it peaks, but that's not the only place. Am I right?
I'm just repeating what these studies say. The studies say 40 hertz.
This is a combination of light and sound while someone's sitting in a chair for
right.
of minutes a day. They're exposed to the same frequency of flashing light and sound, which I thought was a really interesting approach. And they have studies in animals and humans, and they're moving to clinical trials.
Seems like I could go over to Europe and get 50 hertz and just camp out underneath a high tension line and get the same thing, huh?
They found it's not at 50, it's not at 60, it's at 40.
Okay. I didn't know that.
And the unique thing about the studies in 2024 is this woman at MIT, Dr. Tsai, Tsai, she's been working this 40 hertz intervention science for at least a decade now. I saw her in 2017. That 40 hertz reverse cognitive decline. Now we know the mechanism of action. That's where this 40 hertz science is now different. It's not just that we know the effect, we actually know the mechanism of action in the brain responsible for the 40 hertz benefit. And that benefit is in triggering the glymphatic system. It's lymphatic with a G prefix to the word, glymphatic system. If you Google 40 hertz glymphatic, you'll find the studies.
That's right. In fact, I've been working with a local electronics engineer and have made some 40 hertz flashing LED lights, and I've sold a few already. That's Liwei Tsai at MIT, and
Yeah.
she's a very good researcher. And they're also looking at 40 hertz tones as well. It's something that it appears to be fairly easy to make. I haven't done anything with electromagnetic waves, just with flashing lights and with tones.
Right.
What note on the piano is 40 hertz? Josh should be able to tell us.
It's the lowest E. The lowest
E. Okay.
John, have you tried combining them, and can you tell us what type of feedback you're getting from people who use them? I'm mostly curious in protocol questions like how many minutes a day, but critically, have they tried combining light and sound, or are they just getting benefit from light
Well,
alone?
Liwei Tsai has tried combining them. There's some confusion about whether if they go to different parts of the brain and the pathways take longer, it might not really be combined in phase. We've been mostly working just with the flashing lights or the tones separately.
What frequency is the light, or is it just white light?
I'm using white light. It sends a stimulus to the brain with each little flash.
If you go further down the infrared, it actually penetrates through the skull.
No, this goes through the optic nerve.
Okay.
And that's how they did their experiments with the mice, and they're doing their experiments with people now. With the mice, they found that an hour a day for a week reduced the amyloid beta load by 50% in just 1 week in mice that had been bred to develop amyloid beta.
That's such an amazing
Is
finding.
that
John.
astonishing or what?
Are
Right.
your clients being given the same protocol, an hour to start, or are you suggesting shorter periods and working up? What are people liking?
I just turn it on my desk and leave it on for a while. I'm not that precise.
Can you overdose?
I've seen no evidence of that. You can look up Liwei Tsai and read her extensive reporting on the subject.
Most other light therapy studies do show negative effects as you go out past 20 minutes with almost any type of therapy.
For 40 hertz?
Well, I said most light therapies. I haven't seen this question studied in 40 hertz, because unlike all the other therapies where they're trying to give you a constant exposure, or most of them usually, stay away from flashing on any cycle. So obviously 40 hertz is flashing, and that is detectable in a way that, say, a constant LED or a chip on board is not flashing.
Yeah, this is not like the infrared that people are shining on their skulls to grow hair or something like that. This is just not very bright, flashing 40 hertz light.
Does the brightness matter? There must be an optimum somewhere.
Well, it's not like the light is doing something to the brain. It's just entraining the natural gamma brainwave frequency, which is 40 times per second. It's just a matter of entraining the natural frequency of the brain.
Seems to me that we're in early days. it appears that it is the frequency that makes the difference. And the mode that the frequency is taking place within, whether it's light, sound, or PEMF also makes a difference, doesn't matter as much as the frequency per se. I've also been looking, like, what is the significance of 40 hertz? And I did what I always do when I have a question like this. I check out the impact of the M1 muscarinic acetylcholine receptor in the brain. The M1 muscarinic acetylcholine receptor in the brain is the trigger for the cholinergic anti-inflammatory pathway. And that study I just posted a link to, published back in 1998, says that when you ping that receptor, it results in a 40 hertz signal. That's the way I read that study. This is more speculation on my part, but
40 hertz is one of the brain's natural frequencies. It's called the gamma frequency. We have other alpha and beta and
right. Yes. Well, and there are other gamma frequencies.
yeah, the gamma range is from 40 to 100, I believe. But Leeway size research found that 40 was the one that was reducing amyloid beta deposits in the brain, which is correlated with when we sleep deeply and there's more clearance of amyloid beta. I think there's a relationship with the gamma frequency there too.
Maybe we should think about this from an evolutionary perspective. 40 hertz, that's really low. You can wonder how this came about and why it's meaningful.
Well, actually, it's pretty high compared with the alpha frequency and the beta frequency of the brain. So it's not low for brain waves.
Yeah, you can play a tone of 40 hertz on YouTube. There's a 40 hertz test tone if you want to listen to it. I suppose I could just take a nap listening to that and get a lot of the same effect, huh?
Well,
40 hertz is at the limit of about what you can perceive as a tone. You almost hear it as separate thuds.
Yeah.
It's lower than the lowest note on the piano. And when you want to reach those low notes, there's hemisync technology, which puts one tone in the left ear, the other tone in the right ear, and the difference is 40 hertz. So you hear these beat frequencies at the signal you want to stimulate instead of getting the tone directly through the brain through auditory, because it's not an auditory signal. I think 40 hertz is borderline, whether you can perceive it as a tone.
Well, actually, you may not want a tone. You may want a series of pulses 40 times per second. And it could be at any tone. You could have like a 200 cycle tone that flashes briefly 40 times per second. Because that's what we do with the light. The light isn't 40 hertz electromagnetic radiation. It's white light flashed at 40 times per second.
I added a reference in the chat for a study that shows 40 hertz vibration stimulation on the skin also has a positive effect on amyloid beta, and I think that's pretty interesting that our body is able to detect it through all these different pathways.
Yeah, I think they all the nerves go up to the brain, and it's the brain we want to influence.
But again, I wonder how this came about. That's purely speculative, but it seems most everything that's part of you is something that developed over millions of years, and it must be in the environment somewhere that would cause this. I wonder what it is.
No, it's a natural brainwave frequency. We're just stimulating it.
Well, is this the same in, say, mice, or is it just us who have it?
They found it in mice first.
Okay. Oh, interesting.
Wild thing. You can also do a search for REM sleep. Rapid eye movement sleep is associated with 40 hertz. What I take that to mean, and I've never tried to articulate this before, because 40 hertz is associated with REM sleep, Mother Nature actually established a sleep mechanism that automatically triggers 40 hertz. It's too much of a coincidence to understand that sleep is associated with 40 hertz, and 40 hertz is the frequency that triggers the glymphatic system. I said sleep, I mean REM sleep.
Wonder if you do all these things together. While I'm having my young, fresh frozen plasma, I'm also getting stimulated at 40 hertz in my eyeballs and in my vibrations. And oh yeah, let's have some hyperbaric oxygen thrown in there. Wonder if you can have a cumulative, or if it would begin to be toxic.
I'm very skeptical of hyperbaric oxygen. I think it cross-links the tissue in the lungs and probably ages you faster. That's my unresearched opinion.
You're not alone in being skeptical about it. I just mentioned it because it's a common thing that people do. And you could throw an infrared in there too, right?
Hyperbaric oxygen is a profoundly healthy thing to do, along with EWAT. I think for healthy people who can exercise, EWAT is better. But in 2020, Shai Efrati in Israel found that in humans, clinical study, hyperbaric oxygen therapy for, I forget how long, a month, 2 months, something like that, increase telomere length and reduce immunosenescence. And it's been shown that hyperbaric oxygen therapy increases PGC1A.
Has there ever been live study on, say, for instance, rats showing that hyperbaric oxygen has any notable effect on lifespan?
I don't know.
I mean, hard to do it on humans, but it'd be interesting.
It would.
Walt and anybody else who's thinking of trying to combine multiple things at once, if they're drugs, we call it polypharmacy, and it's considered a bad thing to take more than 5 or 10 things at a time. And we encourage clients who need to take 10 or more things a day to space them out, because inevitably they may be combining drugs that would cross-react. And I think the same is true of any other therapies that are known to have profound effects. They may cross-react. That's just one reason to separate them all by a couple hours.
But you don't know. That's
But
just
you can
speculation.
assume that anything that has a profound effect on you will influence the next thing you're taking that's also going to have a profound effect on you, either in a bidirectional sense, as in the hemoxygenase pathway, blood pressure, heart rate, so many bidirectional pathways where something's either going up or down with profound effects when it does so. Whatever you think is most important, you might say, well, I'm going to take all the things that make my heart rate go up together and all the things that make my heart rate go down together, or at least not mix them. You could say the same for blood pressure. I recommend the same for the hemoxygenase universal stress enzyme that's going to boost your carbon monoxide, your iron, and your bilirubin. But however you worry about it, I urge you to worry about it in one dimension or another. The better you control for it. And again, the best way is just separate everything by 3 or 4 hours, and then you'll get a fair shot at what was the reaction I had. The last thing I want to say about it is if you want to know what your stress reaction is to any substance, just measure your basal temperature with a thermometer that has an extra digit. They're $15 at any pharmacy, and you can easily see the small temperature changes that are associated with boosting your hemoxygenase stress pathway. And it may take a couple hours to go up and come down again, or it may take 30 minutes, but you'll get a clear sense in how quickly and how high your temperature goes, how stressful anything you do is on your whole organ. Thank you.
It's a study that was done that combines PEMF and hyperbaric oxygen therapy, and it found that the benefits of doing both at the same time were better than doing either alone. And I have done EWAT, which is a kind of increased oxygenation of the body while exercising. And I've done that and PEMF at the same time, and I feel pretty good at the end of it. But feeling doesn't count. There's a study showing benefit of doing both.
Would I want to go ahead and turn on my infrared lights while I'm in there and maybe also have some of the 40 hertz noise going on?
There's a video showing Ben Greenfield with near infrared light showing on his body while he was doing on a treadmill. Yeah. So yeah, that's the thing to do. I don't have a body you want to see in a video doing that, but
your near infrared supposedly, from what I've read, it's hard to get good information, but supposedly it stimulates energy production. It stimulates the mitochondria to create cellular energy. So you could understand why that would be associated with better exercise results.
Infrared light increases PGC1A.
I got to read up on this PGC1A.
Yeah, we all do. I'm going to post this bet because I could spend the next year looking for a falsifying experiment, and I don't want to spend a year on this. So $50 of Bitcoin per the rules. I'll post this. I'll send it to you. Well, I'm going to post it a couple places. And
okay, good.
Yeah, so.
We've had 2 hours here almost. Should we let it go now? Everybody happy?
Work great.
Yes, I'm happy.
Thank you again, Sven.
Yeah.
Managed to get us talking like crazy here. Beautiful.
I am done for today. From the morning, I was working on this presentations. I was working through these papers non-stop. And so, like, my brain is fried for today, I think.
Sven, can you share your slides? They were really excellent, and perhaps with the whole GRG list.
I'll post this once I edit it. One of the great things is when you spend that much time on something, you learn it so much better than you ever can from just reading it casually. I found that the best way to learn something is to teach it, because when you have to teach
Yes.
it, you realize the holes in your knowledge and you do something about it. Thanks, everybody. I guess I'll, bye-bye.
Okay, have a nice day. Bye-bye.
Thanks again.